the sum total of processes by which a living organism receives and utilizes raw materials for building its structure and maintaining body functions is called nutrition. And the chemical substances that work as raw materials for bodybuilding and maintaining its functions is termed as nutrient. Now let's discuss essentiality of nutrients. Essential nutrients should be absolutely necessary for supporting normal growth and reproduction. And the requirement of the mineral should be highly specific and cannot be replaced by any other element. And the element should be directly involved in the metabolism of plants. It becomes a component of either a structural or functional molecule. The element may additionally have a corrective effect on mineral balance and other soil conditions. There are two types of nutrients, micronutrients and macronutrients. Micronutrients are the nutrients which are required by plants in small amounts, less than 10 millimole per kg, and their essentiality came to be known only when extra pure salts were used in cultural experiments. And macronutrients are those elements which are present in easily detectable quantities, and these are usually involved in the synthesis of organic molecules and development of osmotic potential. Examples of micronutrients are iron, copper, manganese, molybdenum, zinc, boron, chlorine, nickel, etc. And macronutrients are carbon, hydrogen, oxygen, nitrogen, phosphorus, sulphur, potassium, calcium, and magnesium. On the basis of functions, essential nutrients are of different types. Structural elements are generally constituents of biomolecules. For example, carbon and hydrogen are components of all organic substances. Many of them also contain oxygen. Nitrogen is a constituent of all amino acids, proteins, nucleic acids, chlorophyll, oxygen, and phytohormones. Sulfur is also found in amino acids, methionine and cysteine, which form a number of proteins. Phosphorus is present in nucleotides, and magnesium is a component of chlorophyll. Then there are many energy related elements, like magnesium is a component of chlorophyll that takes part in conversion of light energy into chemical energy. Phosphorus is also a component of ATP which functions as energy currency of the living systems. There are many nutrients which act as activator or inhibitors of enzymes. For example, magnesium is an activator of several enzymes of both respiration and photosynthesis. For example, rubisco and phosphoenol pyruvate carboxylase. Zinc is activator of carbonic anhydrase and alcohol dehydrogenase. Molybdenum is required for functioning of nitrogenase enzyme. And there are many other functions like maintaining osmotic potential. So most of the osmotic potential of cell sap is due to inorganic salts. Osmotic potential is required for water absorption and maintenance of cell turgidity. Nitrogen is required in greatest amount. It is the major constituent of proteins, DNA, vitamins, and hormones, and it is required by meristematic and metabolically active cells. Hence, it is very important for cell division, growth, metabolic activities, and photosynthesis, etc. It is absorbed as nitrate, nitrite, or ammonium ions. If its deficiency is there, then generally chlorosis starts from older leaves and stunted growth is observed due to decreased protein synthesis, and cells do not elongate, and slow divisions take place and premature leaf fall is observed. Phosphorus is absorbed as phosphate ions, and it is an important constituent of cell membrane because cell membrane is made up of lipid bilayer, and lipid bilayer has phospholipids, and phosphorus is important constituent of nucleic acids, and phosphorus is required for phosphorylation reactions. So very important constituent for photosynthesis and respiration. And if there is deficiency of phosphorus, we observe stunted growth of plants, leaves they become dull green, and chlorosis is observed with necrosis first in older leaves or premature falling of leaves, and delayed flowering is observed. Potassium is absorbed as potassium ions. It is required by meristematic cells, buds, leaves and root tips. It maintains cation and ion balance and is involved in protein synthesis. It is also required for opening and closing of stomata. It maintains turgidity of cells, and if there is deficiency of potassium ions, chlorosis is observed first in older leaves and then in younger leaves, and marginal or apical yellowing is observed, and internodes become shorter. We observe loss of apical dominance, and loss of cambium activity is also observed when there is deficiency of potassium ions. Calcium is absorbed as calcium ions, and it is required by meristematic and differentiating cells. It helps in formation of middle lamella during cell division, and it is very essential for the formation of spindle during cell division. So if there is deficiency of calcium ions, we observe stunted growth, degeneration of meristematic tissue, especially root apex. Chlorosis, necrosis and curling of leaves is observed first in young ones and then in older parts. Magnesium is absorbed as magnesium ions. It activates respiratory and photosynthetic enzymes. It is involved in the synthesis of DNA and RNA and very essential component of chlorophyll. It helps to maintain ribosome structure also, and if there is deficiency of magnesium, chlorosis is observed first in older leaves and then in younger leaves, and premature leaf falling is observed, growth is reduced, and phloem is underdeveloped. Sulphur is absorbed as sulfate ions. It is very essential component of two amino acids, cysteine and methionine, and it is also important component of several enzymes and vitamins. It is required for chlorophyll formation and growth, metabolism and nodule formation in legumes. So its deficiency symptoms are similar to those of nitrogen deficiency, because it is important component of amino acid, and because of deficiency of sulfur, protein synthesis is inhibited. Chlorosis is also observed first in young leaves and then in older leaves. Iron is absorbed as ferric ions, and it is essential component of proteins involved in electron transport chain, in photosynthesis and respiration. It activates catalase enzyme and is essential for the formation of chlorophyll. If there is deficiency of iron, we observe chlorosis in plants, which appears first in young leaves and then in older leaves. And then necrosis is observed and growth is highly reduced. Manganese is absorbed in the form of manganese ions, and it is required in splitting of water molecule, hence evolution of oxygen, and its deficiency causes chlorosis, and in case of plants we also observe stunted growth, flowers often become sterile. Zinc is absorbed as zinc ions, and it activates carboxylase and is needed in the synthesis of oxygen. It is an important component of activator or several other enzymes including carbonic anhydrase, dehydrogenase, and it is, as we mentioned, required for synthesis of oxygen. Its deficiency leads to stunted growth, and leaves are not properly formed, and several other types of leaf distortions are observed. Chlorosis is also observed during deficiency of zinc. Copper is absorbed as cupric ions. It is associated with metabolic reactions and is involved in redox reactions during photosynthesis and respiration. It is important component of plastocyanin, cytochrome oxidase, rubisco, and many other enzymes. When there is deficiency of copper, we observe apical necrosis of young leaves in plants and growth is stunted. Boron is absorbed as borate ions and it is required for uptake and utilization of calcium ions, also required for membrane functioning and pollen germination. It is required for cell elongation, cell differentiation, and for carbohydrate translocation through phloem. And during deficiency of boron, death of root and shoot tips is observed, loss of apical dominance is observed, and then stunted growth, small size of fruits, and disintegration of softer parts of plants is observed, which leads to stunted growth of plants. Molybdenum is absorbed in the form of molybdate ions and it is required for the activity of nitrogenase and nitrate reductase enzyme, and its deficiency causes chlorosis and necrosis, and leaves fall before maturation. Chlorine is absorbed as chloride ions. It maintains solute concentration and is also required for water splitting reactions. It is also required for cell division and normal production of fruits. So if there is deficiency of chlorine, leaf wilting is observed, chlorosis and necrosis, and stunting of roots and swollen root tips are observed. Critical concentration is that limited concentration of the essential element below which growth of the plant is reduced. As each essential element has one or more specific structural and functional roles, its deficient supply results in appearance of normal signs called deficiency symptoms. The symptoms disappear when the deficient element is supplied. Deficiency symptoms are the changes which indicate element deficiency. There are different kinds of deficiency symptoms like chlorosis, which is loss of chlorophyll, necrosis, which is cell death process, and stunted plant growth. Premature fall of leaves and buds is observed when there is deficiency of certain elements, and cell division is also inhibited. Problems associated with identification of deficient element. Deficiency of any element can cause multiple symptoms, whereas same symptoms can be caused by deficiency of several elements. And moreover, different plants respond differently to the deficiency of same element. Excess of minerals is often toxic, and any mineral concentration in tissues that reduces the dry weight of tissues by about 10% is considered as toxic concentration. Excess of manganese causes deficiency of iron, magnesium, and calcium. This is due to its competition with iron and magnesium for uptake, and it also competes with magnesium for binding with enzymes, and it inhibits calcium translocation into shoot apex. Plants absorb their minerals from the soil through the roots. The most active areas of the root for mineral absorption are the zones of elongation and root hair. The rate of mineral absorption is usually independent of its concentration in the soil, and minerals are absorbed as ions, which are accumulated by the plants against their concentration gradient in the soil. There are two phases of absorption of minerals. In the initial phase, there is a rapid uptake of ions into outer space or free space of the cells. Outer space means intercellular spaces and cell walls. Ions absorbed in free space are freely exchangeable, and in later phase it comprises of cytoplasm and vacuole. In the inner space, the ions are not freely exchangeable with those of external medium, and entry of ions into outer space, that is initial phase, is passive absorption as no energy is required for it, and Absorption of ions into inner space, that is second phase, that is an active process that is requires metabolic energy. Soil acts as the reservoir of elements. It supplies minerals and has many nitrogen fixing bacteria. When these bacteria fix nitrogen, then nitrogen content of soil increases. Soil particles also hold water, and air spaces are there which supply air to the roots, and it also acts as a matrix that stabilizes the plant. Biological nitrogen fixation requires a reducing power like NADPH, FMNH2, and a source of energy like ATP and enzyme dinitrogenase. And this nitrogenase enzyme is responsible for reduction of nitrogen to ammonia. Rhizobium bacteria live in the soil. They are unable to fix nitrogen by themselves. Roots of a legume plant secretes chemical attractants, and bacteria collect over the root hairs. They also release certain factors that cause curling of root hairs around the bacteria. And an infection thread is formed which encloses bacteria, and infection thread grows along with multiplication of bacteria. Then it reaches cortical cells. Then cortical cells dedifferentiate and start dividing. Then root nodules are formed. Nodule formation is also stimulated by auxin produced by cortical cells and cytokinin liberated by invading bacteria. The infected cells enlarge and nodule is formed.